Thanks for letting me come up and talk. They had to bring out the long-haired professor. Here I am, all right? I think I have 20 minutes. I have at least 250 slides, and there's probably a test at the end, so I hope you brought your pencils. So today I want to talk specifically about biological weapons and how we're working on technologies to try to detect them, defend against them, and actually really just try to figure out what the heck people are doing. And I'll talk about the ones you hear about, Crispr, and I'll talk about some of the new technologies that are out there, how we can detect new types of engineering. But I just want to start out and talk briefly about the sovereignty practice I'm a part of. At Noblis, we are a combination of folks like me, who are traditional kind of scientists, who sit in labs, computer rooms, do this type of research. We also support other programs for program management, SME types of supports across nuclear, chem, bio, and explosives. But like I said, I am mostly in the life sciences practice, and we're going to talk specifically about that. Biological weapons are not new. They're actually the oldest form of weapons of mass destruction. They've been used for a long time. Going all the way back, the first record we have right now is 400 BC, but there was likely one even earlier than that. It was easy for people to figure out, as it is today, that you can make people sick, right? You can make them sick by getting them around a sick person. And there's lots of other ways I'm going to talk about in just a minute we can get them sick. This goes on for years and years and years, and the most recent ones we can openly talk about are the 2002 to 4 ricin labs. These were in apartments, right? And so it's very easy to grow these types of things. Every year, there are search warrants served, our law enforcement folks walk into an apartment where someone has built a lab. Let me try to give you an idea of how easy it is. And this was a conversation I was actually just having with my son. When I was in high school in the early 90s, I didn't have a bacterial incubator for a science project I wanted to do. I built one and ran it in my closet. My parents never knew until I went to the science fair and they asked, well, who at school helped you? And I said, no one. I have an incubator in my closet, and I was growing E. coli, right? So for almost a year, they didn't know I had built an incubator out of a light bulb and a toy tub, right? And this was in the early 90s. So imagine what you can do today. I do look in my kids' rooms all the time, though, so because they're from me. So the important components that we need to look at in biology, there's two major areas that we focus on, but there's actually a third one that's emerging that I'll add to this. So viruses and bacterial agents, they make you sick because they produce toxins or they interfere with something that your body should normally be doing. We know a lot of these things. A lot of them we don't know, but these are the types of things that you can then splice into a bacteria that's easy to grow, easy to leave on a countertop, and all of a sudden someone gets sick, right? Biology naturally does this type of work, and we'll talk about how that happens in just a couple slides. We want it stable. We want it to live somewhere so someone can touch it and spread it to the people they interact with. And we need to be able to grow it. Surprisingly, it's actually not super easy to grow everything we find in nature. Many things won't grow at all in the lab. And so we've figured out ways to do it. We've actually been doing that longer than anything else in terms of adapting bacteria and viruses to grow in a lab. And so we're very, very good at that now. The viruses are a little bit different. Most bacteria will get anybody sick, right? If they produce a toxin, they'll grow inside your mouth or somewhere, and it will make you sick. Viruses are different. Our bodies are really good at fighting viruses for the most part. There are certain viruses that can infect people and certain viruses that can't. If you ever hear the term zoonotic jump, that's where scientists are looking at how an animal has a virus that currently can't infect people, can jump. The biggest example of this is the bird flu. Bird flu is super infectious, very, very lethal to birds, all right? Right now, it doesn't jump to humans that often. There's a scientist in Europe who developed a version of the bird flu that can infect humans through genetic engineering. And that led to all kinds of controversy several years ago. So these are the important traits. Horizontal gene transfer is the way that nature does it, and we do it. And so now all of a sudden we run into the challenges of, did nature do it, or did we do it? So identification is the first step of trying to figure out what's going on. We're obviously in flu season, so one of the important things we try to look at is we do genomic analysis to try to map things. There's a technology called biovelocity that was developed in Noblis's research efforts, along with it's used at JPEO. This is a technology that allows us to look very closely at the genome of a bacteria or a virus or a host. We use it for people as well. Why does this matter? Because there are lots and lots and lots of bases. So think about E. coli that I was growing in my closet as a kid, right? So E. coli is about 3.5 million bases. CRISPR, the technology you've read about, can change one of those bases. So think of a book with 3.5 million letters and one letter changed. You have to find it, right? You need to know what that change is. So highly accurate, fast analysis of the genome is critically important in trying to move forward with the research. I'm going to talk about an example of Ebola in just a minute. This is how it works. It's called a Reid Mapper. So if you've heard about modern genetics, next-generation sequencing, that's also known as pyro sequencing. What's done with that? So if you think the early Human Genome Project, they wanted to go one end to the other end, all right? And they said, we're going to figure out every single chromosome of the person, starting at one end, going to the next end. This was called Sanger sequencing. This was the early work in sequencing work. Then came pyro sequencing with J. Craig Ventnor and Celera. So they came up with the amazing idea of, we're not going to do this serially. We're going to copy the genome hundreds of times. We're going to break it into pieces, and we're going to sequence each one of those pieces and put them all back together again computationally. So Reid Mappers are the technology that came out of that. Instead of putting the genome back together, we actually just analyze the individual copies and parts, and we use math to figure out where that single base pair change is and whether it actually affects the way the organism operates. So this is an example of how it actually works. This signature sequencing technology is, like I said, used in a couple places, but most at JPEO we support a program that tries to identify evolutionary drift in important biomarkers. So think about the 2014 Ebola outbreak. That was a challenging one because Ebola, which is already fairly lethal, became even more lethal, right? Which immediately makes those of us who follow this nervous because there's something that has changed in how it's infecting people or in how it's actually causing damage to the cells. So there were a couple ideas that came out right off the bat. Broad, our friends up at Broad and at MIT, thought that it was a brand new variant, all right, which shocked us because that's our job, to make sure that there are no brand new variants that we can't find. So what we did is we ran this pipeline on as many samples as we could get. Surprisingly, in outbreaks, it's difficult to get samples out of countries because countries don't want to admit they've got a problem, you know, like people, right? But we did get as many samples as we could of people who'd actually been infected by the virus to try to understand, was MIT right? Was this actually a new virus? And what it turned out to, from our process in this analysis, which is basically referential based. So this is kind of the point in my talk where I talk about how our bioinformatics and our computational analysis works. Biology for a long time used to be a collector's game, right? You think about early life sciences people, they would go out into the woods, somewhere never been before, and they would pick up a plant and they'd say, this is new, and they compare it to pictures or examples of other plants. They take it back to the museum and look at all the plants that had 4 leaves and say, no, this is a brand new species. We're still doing that, except now we do it computationally. So when I talk about the 3.5 million bases of E. coli, we are literally comparing each one of those bases to every example of E. coli that is in the public or private collection. So E. coli has about 10,000 deposits in the national NCBI, the National Center for Biomedical Information at the Library of Medicine, which is the biggest collection. European Bioinformatics Institute has another collection that's mostly crossed over by those. But so we're comparing it to 10,000 different variants. That's how we figure out whether it's E. coli, whether it's new. So the Ebola virus, we don't have quite that many of Ebola virus, but we certainly have quite a few. And so this process is all about going back to the museum, looking at what we have and trying to understand what's different. And so these are the signatures that existed in 2013 going into 2014 for Ebola virus. They were really based on a couple of variants that we knew were commonly occurring. The sad thing about Ebola is there is an outbreak every year. It just happens. It's seasonal like the flu, right? And we know mostly where it starts and where it goes. 2014 was different because it was very lethal. It went farther than it had gone before, and it started in places that we did not expect it to start. And all of a sudden, our markers weren't working anymore. People who were coming in who clearly had the symptomology of Ebola weren't testing positive with the markers that we had tested knew work. But that's because our library wasn't quite big enough. And so after we did the analysis, we've now created all of these new markers that we have for Ebola virus. And now all of a sudden we can find all of these things. I'm going to speed along to get to synthetic biology. There's an important report that was put out by a national, by the National Academy of Sciences. I should know this. My father worked there. So the concept is, what are the important things that we need to look at in synthetic biology? You can go read it. It's an important document to understand, but the things that it really boils down to is intent. How do we figure out the intent of somebody who has engineered a new organism? And this is the main focus of the Felix program that we support now. I'm one of the principal investigators on that. The goal here is to first identify new engineering that we've never seen before and determine whether that new engineering is something that showed up naturally through normal processes of biology, or it's something that somebody actually created. This is horizontal gene transfer. Our favorite example of this is, again, E. coli. E. coli loves to pick up toxins and genes from lots of different places. But the 2011 German E. coli outbreak's a great example. It picked up a really nasty toxin, and then it sat in storage for a long time, and then they grew up the plants that the toxin was on, and all of a sudden people got sick. This happens all the time. It's happening right now. Somebody in the world is sick from this. But, not this particular example, but some horizontal gene transfer has made somebody sick. This is the easiest. We can do this in the lab. All you have to do is grow E. coli with something else that it potentially wants, and you'll end up with some population of your E. coli that now has that toxin. Horizontal gene transfer is very easy. CRISPR is another one. Sorry, I'm skipping so fast. I got the 5-minute warning. CRISPR is another one where you can, instead of growing things together and try to get that transfer, you can all of a sudden individually splice your new toxin into your organism, or you can knock out a function. Imagine you have a function in a bacteria that makes it difficult to grow in the human body. Those things exist. Well, you can use a CRISPR to put individual edits in the part of the gene that turns that gene on, and all of a sudden the gene won't turn on anymore. And so now you have a new virulent version of that bacteria or virus. This is some of the latest technology that's used. We use natural language processing along with artificial intelligence to try to determine what that intent is. So we look at all of the different components, going back to our library, our museum, and saying, we've seen this gene here, we've seen this gene there, we think it does this, we think it does that. We now turn that into a sentence of, this was engineered to do X. That's where the science is going. We're not quite there yet, but it's a combination of natural language processing and artificial intelligence. That leads me to my last point that I'll just jump into. Biology is becoming a cyber problem, all right? So because we have this great reference database, remember that museum that we've gone back to for years to go compare our four-leaf plant to another four-leaf plant. We're now comparing genes to another gene. That's publicly available. Anybody can deposit data into that. And so now it's highly susceptible to data poisoning. So what's data poisoning? The concept of data poisoning is pretty obvious once you think about it. If you're going to build a machine learning tool that uses a reference library like we do in biology, we'll just put a bunch of junk in your reference library, and all of a sudden the machine learning tool won't work. Think about the natural language processing tools that we're using to try to build sentences about the intent of how someone engineered something. Just throw in a bunch of sentences that don't mean anything. All of a sudden your data doesn't work anymore. I'm going to skip this, come back to it if I have just a minute. This is the concept. If you continue to deposit bad data, and you don't have to purposely deposit bad data. You could deposit bad data because you're a postdoc and it's late at night and you just put in bad information. That's still data poisoning. So one of the challenges we now have in biology is we're going back and trying to clean up those databases so that we now have highly curated databases that we have complete trust, or mostly complete trust, in whether that natural language processing engine or that bioinformatics tool will actually help us identify what we're looking for in the intent. The problem is a lot of examples that we use to say we've never seen this before are gone, all right? And so it's not a biology problem, it's now a cyber problem. How do you protect those databases? How do you make sure that people who actually know what they're doing are curating them and making them high quality and can actually produce the information that you want. I'm going to jump back if I can. I don't know if I can't go back. Yeah, I don't think I can come back. One of the other things that it's used, that natural language processing, that NLP as well as machine learning is used, that's important for data for poisoning, is the latest generation of sequencers. The latest generation of sequencers, they're called Nanopore Sequencers. The biggest producer is Oxford Nanopore Technologies, a British company. It uses a trained artificial neural network to actually determine what the sequence is. So there, you don't even have to poison the reference library. You just have to mess up the way the algorithm works. And all of a sudden, you're going to get a bunch of garbage. So a very clear example of this. There's scientists who have engineered different types of nucleotides. So these are not the normals A, T's, G's, and C's. They have 4 new ones. The natural language processing engine that the new sequencer uses can't see those 4 new bases. So now all of a sudden you have a whole part of the genome that you're completely blind to. If it was in the organism, you would never know it with the new types of sequencers. So, this is a major problem. My goal here was to convince you that biology is becoming a cyber problem. I hope I have. I think I have probably 10 seconds for questions, but some final thoughts, I think I've already hit them. If there is maybe one question, I'll jump into it.
This particular agriculture product, that there is that kind of risk. If you could answer that.
Yeah, so there's, I have 2 answers to that. We'll start with the obvious one, and there's a little one that's a little bit less obvious, which is an area of research we're just starting to get into. The first one is, endemic pathogens just exist, right? And so a great example is it used to be very difficult to raise chickens in Northern California, in the San Francisco Bay Area. It turned into a big issue when that area was growing significantly during the gold rush, right? They had a hard time getting proteins because you couldn't grow chickens because the chickens kept dying from endemic flu-like viruses, and that's just the way they worked. So yes, in California, there are salmonella and other types of pathogens that show up in large farms, industrial farms. Some of it has to do with processes. Some of it has to do with that's just where they are, and we get our food from lots of different places. And so it makes you sick because you're not used to getting food from that place, and that moves me into the second one. There's the new area of research that folks are starting to get into is microbiome. So the human microbiome projects were recently finished at the NIH. Finished meaning they just collect a baseline set of data. There is a syndrome many of us have had that has 2 names. First is soldier's stomach, and the second is traveler's stomach. If you've ever gone to a foreign place and you had a meal and all of a sudden didn't feel so good for a day or 2 or maybe even longer, that's what you got, right? You didn't get food poisoning. Your gut microbiome is highly developed for the local area that you live. And so it may not be that, sure, there may be salmonella on that melon, but most people who live in that area and naturally eat what's grown in the soil there don't get sick from it. But when you ship it to Southern California, you ship it to Virginia, all of a sudden people get sick from it because their gut hasn't adapted to fighting that particular pathogen. And the problem with that is you can't feed them a bunch of antibiotics because it kills their gut microbiome, and all of a sudden it doesn't fix the problem. An interesting statistic came out in a paper maybe 18 months ago by the Naval Research Labs is that the rate, the soldier disability rate for soldiers' stomach during the Afghan and Iraq War was the same as World War II. We don't have a good solution to this. And so that's just it, is it you should eat food that's grown locally if you don't want to get that sick. But the reality is that's not the way our food system works. We're trying to make it better, though. That's it. I don't have any more questions. I do want to acknowledge folks who funded this work. I mentioned JPEO, DITRA, and IARPA. So thank you.